In this lecture, we are going to now cover the third and fourth stages of cellular respiration. That's the Krebs or citric acid cycle, and then the electron transport chain. To finish off the lecture, we're also going to touch on anaerobic respiration and how that differs from aerobic respiration, which is what we are talking about here. So like we said in previous lectures, some of the textbooks that you may have seen combine the pyruvate processing step and the Krebs or citric acid cycle into one process. But what is the Krebs cycle? Well, the Krebs cycle, as you've heard before, was discovered in inverted commas by Hans Krebs, who figured out that there are 8 small carboxylic acids, basically these small organic acids, that are rapidly oxidized in a very set sequence, and that this cycle runs twice. Now, what you need to do as homework from this lecture is to answer the question, why does the Krebs cycle have to, or why does it run twice? And this is something that we will ask in tests and the exams. So essentially what we have happening is that there is the oxidation of the acetyl coenzyme A, which comes from the pyruvate processing, into 3 molecules of NADH, 1 molecule of FADH2, and you should remember what that stands for, and either GTP or ATP, depending on the cell type, or the cell type that the process is happening in, and we'll talk about that in a moment. Right. Let's just remind ourselves that in the previous stage, pyruvate processing, acetyl coenzyme A combines with oxaloacetate to form citrate, and that is the most important, the first substrate required for the Krebs cycle. Okay, it contains 6 carbon atoms, and it's catalyzed by an enzyme called citrate synthase, and that's very important. As we go through the Krebs cycle in particular, it's very important to remember the enzyme that catalyzes the particular step in the reaction. So another homework question for you is to determine and figure out where the oxaloacetate in this stage of pyruvate processing comes from. Where does this come from? This is something that we will ask in tests or exams. Right, so to summarize the Krebs cycle, we're going to end up with a completely filled in table like this. So for each of the 8 stages or 8 steps of the cycle, we're going to fill in what the initial product is, what process occurs, the enzyme that's involved in producing the final product, which we will then put in the last column. Now this diagram or this table is very important to revise, because this is something that we will ask either as a table to complete in a test or an exam, or for you to label and annotate a diagram. So as we go through each of these 8 stages over here, you will have a diagram and some text, and ultimately we will end up with a completed table, all filled in each of these cells, and that will come up towards the end of the lecture. And you should remember to revise this and redraw it in your notes when you are going through the revision of the Krebs cycle and cellular respiration. Right, so let's get underway with step number 1. Step number 1, stage number 1 in the Krebs cycle, up here as denoted by the citrate molecule over there. As you'll see, as we go through each of the stages, they will light up in red as we go around, and we will talk about each of the steps, have some text for each of the steps down at the bottom, and describe exactly what goes in, which enzyme is involved, and then what the final product is that hands over to the next stage in the Krebs cycle. So the first step is that citrate first undergoes dehydration, and the product of that dehydration is then hydrated to form a substance known as isocitrate, as you can see over here. And this process, going from 1 to 2, is facilitated by the enzyme aconitase. Aconitase. So that enzyme is important, as you'll see in the table as we go through. So this is that this isocitrate then is oxidized and decarboxylation occurs to produce carbon dioxide, as you can see over here, and our very first NADH. Remember that NADH is an electron carrier and something known as ketoglutarate, right? And this is facilitated by the enzyme known as isocitrate dehydrogenase, right? And it's very important that we remember that decarboxylation is the removal of a carboxyl group, and that results in the production of carbon dioxide, an important product of cellular respiration overall. In the third stage, that alpha ketoglutarate that was produced in the second step is then undergoes oxidative decarboxylation to produce carbon dioxide. Remember the removal of a carboxyl group, and carbon dioxide is produced. Our second NADH electron carrier and something known as succinyl coenzyme A. And this entire step is facilitated by the enzyme alpha ketoglutarate dehydrogenase. And we will put that all into the table at the end. The fourth step is when this succinyl coenzyme A over there is phosphorylated to produce succinate and either GTP, remember what GTP stands for, or ATP, facilitated by the succinyl coenzyme A synthase. Now, it depends, the GTP or ATP depends on the cell type. So if it is a muscle cell within, let's say, for example, a human body, ATP would be produced. But in a liver cell, GTP would be the product. And what I would like you to do is to go and find out why GTP is produced in the liver cells and not in muscle cells. Step 5 sees the succinate that was produced in step 4 undergoing oxidation to produce our first FADH2 and something known as fumarate. This process is then facilitated by the enzyme succinate dehydrogenase. Then the fumarate is hydrated to produce malate, and this is facilitated by fumarase. Step sees the malate being oxidized to produce oxaloacetate. So there's a little clue as to where oxaloacetate may be coming from, that question that we asked previously or earlier in the lecture. And then the third NADH electron carrier, facilitated now by the enzyme malate dehydrogenase. And then step 8 of the Krebs cycle sees the acetyl coenzyme A combining with oxaloacetate to form citrate. Does that sound familiar? I hope it does, because that's right at the beginning, the end stages of pyruvate processing, 6 carbon atom, and the reaction is then catalyzed by citric acid synthase, and the process occurs again. Right, so here is that summary table, and at this point it would be a very good idea for you to pause the video and then redraw this entire table so that you can see, right, at the beginning, step 1, stage 1, what is the initial product? What process occurs, using which enzyme, and what is the final product that is then moved through to the next stage? Note, very importantly, the number of electron carriers that are produced, how many carbon dioxides are produced, and how much ATP is produced at each step, because that will be important in the overall summary of cellular respiration, and we will get to that in a moment. Right. So it's important to remember that all three of the processes that we have discussed so far in cellular respiration are quite tightly regulated. Certainly in pyruvate processing, the Krebs cycle and glycolysis, as you've heard, when we have a lot of ATP and a lot of NADH, so NADH, the electron carrier, in the cell, it implies that the cell has sufficient energy for its functioning. And so the overall process of respiration is actually turned down or deregulated. Remember, we were talking in previous lectures about negative feedback loops, and this is a good example. When there's a lot of ATP and NADH, as you can see over here in the diagram, nicely summarized, it turns down, or there's a negative feedback in the overall process of respiration. By contrast, when the cell has only a little bit of ATP, so basically a lot of ADP, and only a little bit of NADH, so lots of NAD+, it means that the cell has got a low energy state. So respiration as a process overall is turned up. And so that's how these processes, certainly the first three stages of cellular respiration, are regulated within a cell. So, what have we done so far? We've done the first three stages of cellular respiration, and the most recent one being the Krebs cycle, where we saw 2 acetyl coenzyme A's going in, along with 6 NAD+, 2 FADHs. What comes out is we have 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 either GTP or ATP being produced, depending on whether it's a muscle or a liver cell. And this process happens within the mitochondrial matrix, much like pyruvate processing, but outside of the cristae. You should remember what the cristae are within a mitochondrion. Right, so what have we used so far in the process of respiration, and what have the products been? Well, for each glucose molecule, we can see that we have had 2 NADHs being produced and 2 ATPs. Remember, in glycolysis, 4 ATPs are produced, but 2 are used in the overall process. We have 2 pyruvates, which then go into the mitochondrion. 2 carbon dioxides are produced along with 2 NADHs. Acetyl coenzyme A is produced, and we then have the Krebs cycle, which runs twice to produce 6 NADHs, 2 FADH2s, 4 carbon dioxide molecules, and 2 ATPs. So overall, we are looking at the products being 6 carbon dioxides, 10 NADHs, 2 FADH2s, and 4 ATPs after the first 3 phases of of cellular respiration. So what then happens during the final stage of cellular respiration? The final stage of cellular respiration, as many of you will know, is what's known as the electron transport chain. And the summary equation for what we have thus far is provided above. And what happens, here is the text, is that our electron carriers, the NADH and FADH2s, are used to power a mechanism across the cristae of the mitochondria, using an electron transport chain to produce many more ATP molecules for the cell. So we have both these mobile, and the mobile ones are known as ubiquinone, and immobile, so stationary electron carriers that are embedded within the mitochondrial membrane, within the cristae. And electrons, using our electron carriers, NADH and FADH2, are transported across these membranes from one carrier to another. And then the final acceptor of the electron is oxygen. And that's why water is produced at the end of it. But the key point, folks, is this entire process produces many more ATP molecules than the previous 3 steps in cellular respiration combined. So let's have a look at this in a little bit more detail. So what we have, if we just look at the diagram for a moment, we have the intermembrane space, so between the inner and the outer membrane. We have the mitochondrial matrix, so within the inner membrane, the intermembrane space, so over here would be the outer membrane, if you imagine it being there. So what we have within the matrix, remember that the Krebs cycle occurred with a Krebs cycle, and the process of pyruvate processing occurs within the mitochondrial matrix. And here we have, just for diagrammatic purposes, several of the complexes that are used to transport electrons. Okay, and so what we have is we have electrons that are transported via these complexes, and energy is provided for protons to be pumped across the membrane, so into the intermembrane space. And this proton gradient is used to power a very important enzyme known as ATP synthase, where ADP and inorganic phosphate are combined through the process of oxidative phosphorylation. Remember, oxidative is important because it's the final, oxygen is the final electron acceptor to produce ATPs. So the production of ATP through this process is via this proton gradient. So the protons are pumped across the membrane, okay, and this whole process is called chemiosmosis because it requires the enzyme ATP synthase, which is down here in the diagram. And when the protons are passed through the membrane, it spins this knob over here, which allows the ADP and the inorganic phosphate to combine to form ATP. Now what you should do is you should check on Moodle and have a look at the video by Bozeman Scientific, which will give you a really nice animation of how this process works. But the bottom line is that there is a gradient of protons. So in the intermembrane space, there's a high proportion of protons that are then pumped across into the matrix using ATP synthase, ATP is produced. So after all of that, the 4 stages of cellular respiration, what do we have? Well, when it comes to electron transport, we know that the products of the Krebs cycle, 10 NADHs, 2 FADH2s, and oxygen go in. This produces approximately, as you'll see in the next slide, approximately 25 molecules of ATP, so much more than all 3 of the previous stages, and water. And it occurs in the membranes of the cristae of the mitochondria. So, one of the last questions we need to ask is how many ATPs then are produced from one glucose molecule? And textbooks and the internet will range between 36 and 38. In the table that you saw on the previous slide, there were actually 25. It's approximately 29 or 25 according to that table. But theoretically, there should be a yield of 38 ATP molecules for each glucose molecule during respiration. Okay. However, there are usually losses along the way. Remember, there's a cost for moving the pyruvate during the process of glycolysis. There's costs of moving phosphate, ADP, the substrate for ATP synthesis into the mitochondria. So remember, this is not, you have to spend energy to make energy. And all of these substances are actively transported across membranes using carriers that utilize stored energy and using the electrochemical gradient. So realistically, the number of ATPs that are produced from one glucose range between 28 and 30 after all 4 stages of cellular respiration have been completed. So don't forget this final formula. Glucose plus oxygen gives us carbon dioxide, water, and energy in the form of ATP. Summarized very nicely in this little diagram over here. And you need to remember that it's important to know, based on that table that you've been doing, how many of each go in and how many come out. Right, so far we've been talking about aerobic respiration. What happens when we don't have any oxygen? Well, the short answer is that the process continues, but there are different acceptors of the various products in the stages of cellular respiration, which result in different final products being produced. And this has implications both for the functioning of cells and also in certain biotechnology fields. So let's have a look at the process of anaerobic respiration. So when oxygen is not present, the electron transport chain cannot operate, okay? Because it is important to remember that oxygen is the final acceptor in the electron transport chain to produce water. So electron transport cannot happen when oxygen is not present, okay? So we have NADH plus being produced from the NADH. And what we see is that glycolysis continues even when the final electron acceptor, oxygen, is not present, all right? But pyruvate, or a molecule that is derived from it, as you'll see in a moment, accepts the electron from the NADHs. And in humans and most mammals, this is in the form of lactic acid. You'll see that in a moment. But in bacteria, which is important biotechnological implications, that acceptor is ethanol. So ATP is still produced through the process of substrate phosphorylation, but what we see happening certainly in bacteria is we see now, instead of aerobic respiration, we see fermentation occurring. So let's have a look at bacteria. And this happens in bacteria, also in yeasts, and the ultimate product is alcohol. And this is something that is very important in, obviously, the beverage industry, production of drinks like beer and wine and so on, where you have the glucose being converted into pyruvate. But then what we see happening is that instead of oxygen being the final electron acceptor, that electron acceptor, final electron acceptor, becomes ethanol. And so alcohol is the final product of this process. By contrast, in humans and most other mammals, the final product or acceptor is lactate, which produces lactic acid. And this is actually toxic in the cells, or the muscle cells in particular of mammals. And this is actually, if you are somebody who exercises frequently, this is actually the burn that you feel when you undergo activity like climbing steps or going for a run and so on, where there's no oxygen, or there's very little oxygen in your muscle cells, and so it's converted into lactic acid. And this is actually the burn or the toxicity that you feel after this process of strenuous activity. So during exercise, what happens in our muscle cells is that the oxygen that is available is actually consumed faster than what is available. So the cells have to carry on producing ATP, and this is through the process of glycolysis. But because there's a lack of oxygen, lactate then builds up in the muscles. And when this builds up, the pH of the blood drops and your muscles then become tired. So even at rest, so what happens then when you stop is that this lactate has to be, or is then converted back to pyruvate, okay, because the oxygen debt in effect is paid back. That's why you continue to breathe quite heavily. after you've done exercise. So if you are quite fit, that period of heavy breathing will be shorter than somebody who is unfit, who takes longer to pay back the debt of the oxygen that was not available during the exercise that they undertook. So it's important now at this final stage to remember that yes, energy can continue to be produced during fermentation, lactic acid production or alcohol production, but that's about 2 ATPs worth, okay? But aerobic respiration produces far more, up to about 30, as we saw in those previous slides. And that points to the fact that the evolution then of an oxygen-rich atmosphere was very important in the evolution of aerobic respiration. And this, ladies and gentlemen, has been absolutely vital to the diversification of life. So without aerobic respiration, we may not have seen this diverse range of life forms that we see on Earth today.